Today we're going to be moving on to a new topic, which is the control of gene expression. The way we're going to cover this topic begins with a review of the central dogma. We're going to look at the process of transcription in detail as a review. We're going to define some very important terminology, beginning with the definition of a gene, and continuing into some of the terminology that comes out of genetics to help describe the processes of gene expression. Then we'll move on to some specifics of the global mechanisms that help a cell regulate gene expression. And remember, when we talk about gene expression, we're talking about how a gene is used by the cell for the processes of transcription and then translation. We're going to talk about some of the very specific global regulatory control mechanisms of transcription. And by that term global, I mean the kinds of mechanisms that all cells can use. All cells in all different organisms across all three of the domains of life. Then we will move on to talk about their other ways that a cell can control gene expression outside of transcription. So the things that happen after a transcript is formed, an mRNA is formed, all the way through to when a protein is formed, the steps that a cell can take to regulate that process. And then finally we'll touch on a couple of the cellular control mechanisms that exist that impact gene expression. So we've talked about the central dogma several times, but it's worthwhile to review it again because it's such an important concept in biology. Remember that the dogma tells us that DNA is transcribed into RNA, RNA is translated into protein, and it's protein that creates traits. Now, for some of the genes in the DNA molecule, this process of transcription and translation goes on just about continuously. So for certain genes, we say that they are constitutively expressed. In other words, they're just expressed all the time. They are used for transcription and translation virtually continuously. Another way of thinking of that is saying that these are genes that are always on. And I put the word on in quotations here because, in truth, very few genes are ever completely off if they're being used by the cell. It's more a question of whether or not they're being transcribed more or whether they're being transcribed less at any one particular moment. Remember that most genes inside the cell are not constitutively expressed. Most genes are transcribed and then translated only when the cell has a very specific need for a particular protein at that moment. This is what we call regulated expression. So this is the process, again, of turning genes on and off depending on the immediate needs of the cell. So the first thing we need to do is dust off the details of this process called transcription. And I know everybody learned this process in their previous classes, but we're going to hit on the key points because these are the things that are critical for our understanding of how this process is regulated. So remember, transcription occurs in 3 steps or 3 phases. The first one is referred to as initiation, the second one is elongation, and the third one is termination. And we're going to talk about each one of these phases as they occur in bacteria because it's a simpler model for us. So in the bacterium, when transcription gets initiated, we're talking about the RNA polymerase enzyme getting things started, and it's only a part of the RNA polymerase enzyme. Remember, this enzyme has 2 components that join together to make the enzyme functional. The first one is called the sigma, that's the regulatory subunit, and the second is called the core, that's the catalytic subunit. So this regulatory subunit that we call sigma is the part of the enzyme that binds to the promoter and also binds to the core and brings everything together on the DNA. Now, in order to understand where sigma binds, we need to review how we number bases in DNA. the first base that sits in the coding region of the DNA, in other words, the region of the gene that codes for protein, that base is designated as the +1 site. So any base that comes before that, to the left of that, if you will, we refer to as an upstream base. And we give those bases negative designations like -10 and -35. So when sigma binds to the promoter of a gene, it's binding upstream or to the left of the beginning of the coding region of the gene. And in bacteria, it binds to very specific locations in a very specific promoter, about -10 and -35 bases upstream from the coding region. Okay, now once sigma has bound and has brought the core into position, the double helix of DNA is going to open. Now, the sigma and core of RNA polymerase are enormous proteins. The core, in particular, is large, and it has empty channels or tunnels in it, just like the empty tunnels we talked about when we talked about the ribosome during translation. So what's going to happen is sigma is going to help get the template strand threaded into one of the channels in the core of the enzyme. Remember, when we're talking about transcription, there's only one strand of DNA that's going to be used as the template. That's what we refer to as the template strand. The RNA molecule that gets produced during transcription will be made by matching complementary bases to the template strand. So sigma helps get that template strand positioned correctly by feeding it through one of the channels or tunnels inside the tertiary structure of the core. Now there's a second tunnel in the core of the RNA polymerase enzyme that's the location where the activated RNA monomers are going to come in, all ready to be added on to the growing RNA strand. Remember, in both DNA and RNA synthesis, the bases that get added on to the growing strand have to be activated in order to be used to build a phosphodiester bond. And the way these bases get activated is by having extra phosphate groups added onto them. So in the case of RNA, they become nucleoside triphosphates, or NTPs. Once the template strand is in place, and once the NTP channel is open and ready to function, we're all ready to begin this process of transcription, and sigma gets released. In this drawing, you can see the RNA polymerase enzyme shown in blue, and you can see the DNA molecule shown in gold. So they're indicating in this picture that the RNA polymerase enzyme is large. Like a lot of proteins, it's a very large tertiary structure that you're talking about, that you're dealing with. So you can see that the DNA molecule double helix has been opened in a particular area, and that only one strand, the lower strand in this picture, is being transcribed. So the darker blue area represents the channel inside the enzyme where the active site is, where the incoming NTPs are going to be added on to a growing RNA strand in the order that's complementary to the order of the bases in the DNA. So we have one channel where the template DNA is residing. There's another channel in this drawing to the right where the activated NTPs are coming in. And then there's a third channel to the lower left where the growing RNA molecule, the new RNA that's being transcribed, is going to be exiting the enzyme as it's being produced. So, RNA polymerase has a sigma subunit and a core subunit, but it's the core subunit that's catalytic. It's the core subunit that does the work. of making a new RNA molecule. And it does that by bringing together template DNA with activated monomers called NTPs. So the template DNA sits in one channel, the NTPs enter through another channel, and the brand new RNA that's being made exits through a third channel. Now there's another structural feature of the core of RNA polymerase that we need to remember, and that's the fact that inside the tunnel where the template DNA is being threaded, there are pieces of the wall of the tunnel that jut out into the tunnel space. These are just portions of the protein, the core protein, that have been folded in a way to allow them to jut out into the space of the tunnel. These structures are called the zipper and the rudder. The zipper is the portion of the core that helps open the DNA helix as the RNA polymerase moves along the template strand. And the rudder of the core helps guide the template in the right direction and the non-template strand in a different direction. Okay, so once sigma has the core in place on the promoter and has the template strand in its correct channel, sigma gets released. Sigma has done its regulatory job. At that point, initiation has ended and we move into elongation, which is the second step of transcription. So remember, the base on the template DNA, that's the first of the coding bases, gets designated as the +1 site. So this is where the first RNA base will be placed in a complementary fashion. And then, according to the next base in the template DNA, the next complementary RNA base will be placed. And this continues all along the template strand, building a brand new RNA strand in a complementary manner. Now remember, the new RNA bases are being covalently bound to each other. The complementary binding that goes on between the DNA and the RNA is just hydrogen bonding. But the bonding that goes on between the RNA bases, between, in this picture, the C and the A and the U and the C and the C and so on, the complementary binding that goes on between the monomers of RNA is phosphodiester bonding. These are covalent bonds that join the monomers. Remember too that making RNA requires the enzyme move in one direction and one direction only. We saw this with DNA as well. The cell can only produce RNA in the 5' to 3' direction. That means it can only add on a new RNA base to the 3' end of the growing RNA molecule. Elongation continues along the template DNA until the RNA polymerase enzyme comes upon and creates a portion of the RNA that will cause termination. So on the DNA, there's a sequence of bases that's called a termination signal. It looks just like ordinary DNA, but as it gets transcribed into RNA, that segment of RNA will loop back on itself. Remember, RNA has a tendency to do this. That's how the secondary structure of mRNA forms into a hairpin loop or loops. So the DNA is where the termination signal is, and it's just a sequence of bases. But as the RNA is made from that termination signal, it will fold back over on itself. That loop of RNA will not fit correctly in the channel of the enzyme, and it will cause the enzyme to move away from the DNA, terminating the process of transcription. In this picture, up at the top, you can see the enzyme binding to the promoter of the DNA, In the middle panel, you can see that a string of DNA has been opened up and used as a template. And in the bottom panel, you can see the mRNA that's produced has started to bind back on itself and produce a hairpin loop. That hairpin loop that gets made as a product from the termination signal on the DNA, that loop does not fit well into the enzyme, and so the enzyme lets go of it and transcription terminates. Now, we've been talking about the process of transcription using the simplest model we have, which is bacteria. Remember that when we talk about transcription in eukaryotic cells, we're talking about a more complex process. First thing to remember is that there are actually 3 polymerase enzymes in eukaryotes, not just one, like we see in the prokaryotes. It's RNA polymerase II that's responsible for transcription of mRNA. There are other versions of the RNA polymerase enzyme, including RNA polymerase I, which transcribes rRNA, and RNA polymerase III, which transcribes tRNA. And unlike what we see in bacteria, in eukaryotes there is no single promoter. There is no single string of bases that the RNA polymerase enzyme recognizes as a binding site. Instead, there are multiple promoters. Remember, too, that there is no sigma portion of RNA polymerase in eukaryotes. Instead, there are multiple proteins called basal transcription factors that essentially function the same way sigma does. It's this group of basal transcription factors that locate and bind to all these different promoters and help guide the core of the enzyme into place. Another thing to remember in eukaryotes is that transcription ends with an mRNA that needs to be processed. And in eukaryotes, it's heavily processed. Remember that there are introns and exons in the mRNA. And that means that the introns have to be removed, they have to be spliced out, and the exons have to be joined together. In eukaryotes, the machinery that performs this splicing of the mRNA molecule is called a spliceosome. A spliceosome. And the spliceosome is composed of a group of proteins called SNRPs and a group of catalytic RNA that are called ribozymes, as well as some additional proteins. So the spliceosome is an enormous machine that does the work of splicing out of the introns. We don't see that in prokaryotic organisms. And even after the introns are gone and the exons are joined together, there is still more processing to do. The mRNA has to be capped with M7G, and a polyadenine tail has to be placed on the mRNA. It's only then that the mRNA is ready to leave the nucleus and move into the cytoplasm of a eukaryotic cell. All right, so those are the details of transcription that we need to dust off in our memory. What I want to do now is move forward into gene expression. I want to move forward more into this concept that genes are there for a reason. They're there to be transcribed and translated. And the gene is only one of many that sits in the DNA molecule. Remember that the DNA inside of your cells, each and every one of your cells, contains all of the information that's necessary to make all of the RNA that you would need to make all of the proteins to construct a brand new you. So it doesn't matter if it's a liver cell or a skin cell or a brain cell or a kidney cell, it has the entire DNA molecule in it. Okay? Now that seems strange to us because each cell is so different. And the reason each cell is different is because even though it has the entire DNA molecule in it, it doesn't use all of the genes inside that DNA molecule. In other words, it doesn't express every gene inside the DNA molecule. So the information to make proteins is just the beginning in a life of a cell. All cells have all the information to make every protein, but that doesn't mean they're going to make every protein. In other words, just because a cell has a gene doesn't mean that cell is going to express that gene. It doesn't mean that protein is going to be produced from that gene. There are some genes that are never used by cells. The DNA is there, but it's never expressed. There are other genes that are only used during certain times during the lifetime of a cell. For example, there are genes inside of you that are only transcribed and translated when you were an embryo, and then they will never be used again. There are genes that are only transcribed and translated during infancy, during childhood, at puberty, if you're a woman, during pregnancy. So it's very common for genes to be used at very specific stages in the life of the cell and the life of the organism. The bottom line is that the production of protein is tightly regulated by cells. The cell needs to control whether it makes a particular protein, and if it does make that protein, when it will make it and how much it will make it. So let's take a step back here and remind ourselves about the definition of a gene. What exactly is a gene? Most of us remember that a gene contains a sequence of bases that gives the instructional code to make a protein. One gene, one protein is the rule in eukaryotes. But genes also contain sequences of bases that are used purely for regulation of when that protein gets made. These are stretches of bases that we call promoters and operators and enhancers. They typically reside upstream from the protein coding portion of the gene. So we have genes sitting in DNA, and at certain times in the life of the cell, those genes need to be accessed, they need to be expressed, they need to be used to make RNA and then protein. Now, this all begins with the cell being able to access the gene. And in order for us to visualize that, we need to remember what DNA looks like most of the time when it's sitting in a cell. We tend to imagine DNA in its double helix form, but in actuality, most of the time, the double helix is actually wrapped around histone proteins and tightly packed into the cell. The functional unit of this folded, wrapped DNA is called the nucleosome. Remember, the double helix wraps twice around a group of these histone proteins, and then it goes and wraps twice again around the next group of histone proteins, and then it wraps twice around the next group of histone proteins, and it creates what we call beads on a string. This is each one of these groups of DNA wrapped around histones is referred to as a nucleosome. So you can think of a nucleosome as being the fundamental unit of chromatin, and it's chromatin, DNA plus histone protein, that makes up the chromosome, which is how DNA lives most of the time in a cell. In this image, if you start at the lower right, you can see a drawing of the double helix. And then you see that that double helix wraps twice around that tan-colored circular structure that represents a group of histone proteins. And then it moves and wraps twice around the next group of histone proteins, and then so on, and so on, and so on. Each one of those groups is referred to as a nucleosome. And those nucleosomes then stack one against the other in a very dense formation that we call chromatin, that ultimately makes up what we call the chromosomes. Now you'll notice also on this drawing that they've shown us that the stretch of DNA between each histone cluster, that stretch is there, and it's got a smaller histone circle drawn on it. The stretch of DNA between the two histone clumps, that stretch of DNA is often referred to as linker DNA, and the small histone protein that sits on the linker DNA is referred to as a spacer histone. Just some more terminology for us. So, we know that DNA spends most of its time tightly folded and packed with histone proteins. We also know that gene expression is going to vary from cell to cell. So depending on which genes the cell needs to transcribe and translate, it's going to need to get access to them. Remember, different cells have different functions. So the population of proteins that cell needs at any particular time is going to be different from the population of proteins that another cell needs at any particular time. So gene transcription, gene expression varies from cell to cell. Only the genes that code specifically for the proteins that cell needs at that moment are going to be transcribed. So let's imagine the example of a heart cell versus a cell in the pancreas. The heart cell has no need to express the gene that codes for the hormone called insulin. The pancreas cell does. The pancreas is responsible for producing and secreting insulin. So the question is, if both the heart cells and the pancreas cells have the DNA that contains the gene that codes for insulin, how come the pancreas cell is the only one that expresses that gene? What is it that prevents the heart cell from expressing the insulin gene? That brings us right back to the tight packing of the genetic material inside of a cell. In order for the pancreas to get at the insulin gene, there has to be a regulatory mechanism in the pancreas cell that allows it to unwind or loosen up that tightly packed DNA in the region of that gene. And that same regulatory mechanism has to prevent the heart cell from doing that. So, before we get into the details of this, remember there are some genes that get expressed all the time. Those are the genes that are constitutively expressed. We often refer to them as a group as housekeeping genes because they make the kinds of proteins that all cells need all the time. There's an interesting note on this slide that it turns out that we do have one place in the human body where we don't find any constitutive gene expression, and that's on the Y chromosome. That's just a little bit of trivia for you. The Y chromosome, which evolutionary theorists believe has been shrinking for millennia and will probably continue to shrink over the next several millennia. The Y chromosome doesn't perform any constitutive gene expression. If you imagine the kinds of proteins that a cell would need all the time, that all cells would need all the time, you're talking about things like the polymerase enzymes. We just talked about RNA polymerase. All cells perform transcription, and they do it routinely. They do it around the clock, so all cells need a steady supply of RNA polymerase. All cells are going to wrap up their DNA into chromosomes around the clock. So all cells need to transcribe and translate histone proteins. So constitutive expression can't be forgotten about. We have to keep it in the back of our heads. But remember, most genes have their expression regulated. They only transcribe and translate according to their need at a particular time. And those genes have to be regulated using mechanisms that will prevent sort of accidental or unintentional transcription from occurring. So we're going to talk about what are called these global regulatory mechanisms that control gene expression. And remember, that word global just means that all cells use these mechanisms. And it all starts with the DNA in the nucleus of the eukaryotic cell being tightly wrapped around histone proteins in these nucleosomes. It turns out that we can talk about two very different states of chromatin. We can talk about these nucleosomes being in two very different states of how tightly wrapped they are and how tightly packed they are. The first state is called heterochromatin, and the second state is called euchromatin. When you hear this term heterochromatin, what's being described to you is DNA that is very tightly wound and very closely associated with its histone core in the nucleosome. And therefore, it's essentially transcriptionally inactive when it's in heterochromatin form. Euchromatin is very different. When DNA is in its euchromatin form,